On the 11th of January, 2022, the American Red Cross did something it had never done before in its entire 85-year history of collecting blood. It declared a national blood crisis. Hospitals across the United States were running dangerously low on supply, and doctors were being forced to make agonizing decisions about which patients would receive blood transfusions and which ones would have to wait. Surgeries were delayed. Emergency rooms were stretched to breaking point. And at the very center of that crisis sat one single blood type, the one doctors reach for first when a patient is bleeding out on a gurney and there's no time left to run a test, the one type that can be given safely to almost anyone on Earth in a genuine emergency. Type O negative. And here's the truly unsettling part. This was not a one-time event. In the summer of 2026, the Red Cross declared a national blood crisis for a second time in its entire history, once again driven specifically by a dangerous shortage of type O blood, with supplies of O positive dropping to less than a single day's worth of national inventory, and O negative, the rarest and most universally needed type of all, becoming harder and harder to find. Now stop and think about what that actually means. Somewhere in a hospital right now, a car crash victim, a woman in the middle of a dangerous childbirth, a soldier wounded on a battlefield, or a stranger you will never meet, is depending entirely on whether enough people carrying this one specific, genetically rare blood type happen to walk into a donation center that week. Type O negative blood accounts for roughly only 6 to 7% of the population in the United States, and in some parts of the world, it is almost vanishingly rare, existing in less than half of 1% of certain populations. And yet, when doctors have seconds, not hours, to save a life, this is the blood type the entire modern medical system quietly depends on. So where does this blood type actually come from? Why does it exist at all, biologically speaking? And why, after more than a century of modern genetics, are scientists only now finally uncovering the real, deep genetic truth behind it? That is exactly what we are going to dig into today, and by the end, you're going to understand your own blood type in a way you almost certainly never have before. Think about how strange it is that in an age of gene editing, artificial intelligence, and spacecraft landing themselves back on Earth, one of modern medicine's single biggest vulnerabilities still comes down to something this basic, whether enough ordinary people happen to roll up their sleeve at a donation center that particular week. There is no factory that manufactures O negative blood at scale. There is no synthetic substitute sitting on a hospital shelf ready to go. Despite everything modern science has achieved, the entire system still depends, almost entirely, on human generosity and a genetic accident buried inside roughly 1 out of every 15 people on the planet. And, as you're about to find out, understanding exactly why that genetic accident exists at all takes us on a journey through more than a century of scientific history, a surprising amount of internet mythology, and some genuinely cutting-edge research that is still unfolding right now, in real time, as recently as this year. Let's start with the basics, because understanding exactly what O negative blood actually is requires untangling 2 completely separate genetic systems that most people mix up without even realizing it. Back in 1901, an Austrian scientist named Karl Landsteiner discovered the ABO blood group system, work that would eventually earn him a Nobel Prize. Landsteiner found that human blood could be sorted into different types based on tiny protein markers called antigens, sitting on the surface of red blood cells. If your cells carry the A antigen, you are type A. If they carry the B antigen, you are type B. If they carry both, you are type AB. And if your cells carry neither the A antigen nor the B antigen at all, you are type O. That single letter O, quite literally, stands for the absence of these particular markers. Then, decades later, in 1937, scientists discovered an entirely separate blood classification system layered on top of the ABO groups, the rhesus factor, more commonly known today as simply the Rh factor, or the plus and minus sign you see attached to your blood type. This system is based on a completely different protein called the RhD antigen. If your red blood cells carry this protein, you are Rh positive. If your cells do not carry it at all, you are Rh negative. Put those 2 separate systems together, the ABO group and the Rh factor, and you get the 8 major blood types most people are at least vaguely familiar with. A positive, a negative, B positive, B negative, AB positive, AB negative, O positive, and O negative. Type O negative, then, is simply the specific combination of having neither the A nor the B antigen, and also completely lacking the RhD protein altogether. Here's why that particular combination matters so enormously in a medical emergency. When doctors need to give someone a blood transfusion, the single biggest danger is triggering a severe immune reaction, the patient's own immune system recognizing a foreign antigen on the donated blood cells and violently attacking it, sometimes with fatal consequences. Because type O red blood cells carry neither the A nor the B antigen, and because Rh negative blood carries no RhD protein either, this specific combination of red blood cells has essentially nothing on its surface that a patient's immune system is likely to recognize as foreign, regardless of which blood type the patient actually happens to be. This is exactly why O-negative red blood cells have earned their famous nickname, the universal donor, the one type of blood that can be transfused into patients of virtually any blood type in a genuine emergency, without doctors needing to first confirm exactly what the patient's own blood type actually is. When every single second counts, that fact alone can be the difference between life and death. Think about exactly how this plays out inside a real emergency room. A patient arrives unconscious, badly injured, bleeding heavily, with no medical records immediately available, and no time to draw blood and wait 20 or 30 minutes for a lab to confirm their exact type. In that scenario, doctors cannot afford to guess wrong. Giving someone the wrong blood type can trigger a violent, sometimes fatal immune response within minutes. Essentially, the body's own defenses attacking the transfused blood as if it were a dangerous foreign invader. O-negative blood sidesteps that entire risk almost completely, which is precisely why it gets loaded onto medical helicopters, kept on hand in trauma bays, and carried into combat zones specifically for these exact worst-case scenarios, situations where there simply isn't time to do things the normal, careful way. But here's something that surprises a lot of people, and it's worth clearing up directly because it gets misunderstood constantly online. O-negative is only truly universal when it comes to red blood cells specifically. When it comes to blood plasma, the liquid component of blood, the exact opposite is actually true. Type AB plasma, not O-negative, is considered the universal donor plasma because it contains neither anti-A nor anti-B antibodies that could attack a recipient's own blood cells. It's a small but genuinely important distinction, and one that even shows up in real hospital protocols and disaster relief blood donation drives, where medical teams specifically call out both O-negative whole blood or red cells and AB plasma as two entirely separate emergency priorities. Now, let's get into where this whole story starts turning genuinely strange, because for over a century, the sheer rarity and the almost eerie universal compatibility of Rh-negative blood has attracted an enormous amount of speculation, some of it grounded in real science, and a surprisingly large amount of it wandering off into some genuinely wild territory. One population in particular became the epicenter of this fascination, the Basque people, living in the mountains along the border between Spain and France. The Basques carry the highest concentration of Rh-negative blood found anywhere in the world, a fact first noticed by researchers decades ago, and combined with the fact that the Basque language is a linguistic isolate connected to no other known language on Earth. This led to a wave of increasingly exotic theories throughout the 20th century. Some researchers speculated that Rh-negative blood was connected to unbroken Cro-Magnon ancestry, dating all the way back to the very first modern humans who ever set foot in Ice Age Europe. Other, far more fringe voices took things even further, suggesting Rh-negative blood represented an entirely separate branch of humanity altogether, one that had somehow interbred with the rest of the human population coming out of Africa. And in the more sensational corners of the internet, this theory has been stretched even further still into claims involving ancient astronauts, hybrid bloodlines, and secretive lineages hiding in plain sight. It's worth pointing out just how far some of these ideas have traveled outside the world of serious science. Entire books, documentaries, and countless hours of online video content have been built around the idea that Rh-negative individuals possess unusual physical or even psychic traits, ranging from claims about unusually high intelligence to elevated body temperature to an inexplicable resistance to certain diseases, none of which have ever held up under any kind of rigorous scientific scrutiny. And this extraterrestrial version of the theory runs into an even more basic problem the moment you actually think it through. If Rh-negative blood really did originate from some kind of ancient hybridization event with a genuinely alien species, that species would almost certainly have evolved on a completely different world, under completely different conditions, meaning its biology would likely be built on an entirely different genetic framework altogether, not necessarily DNA at all, and definitely not anything capable of producing a viable, fertile hybrid with an ordinary human being. It's the kind of idea that sounds thrilling in a late-night internet rabbit hole, but collapses almost immediately under even the most basic scientific examination. And let's be very clear about what modern genetics actually says about all of this, because the real explanation, while less dramatic than an alien bloodline, is genuinely fascinating in its own right. Rh-negative blood exists because of a specific, well-documented genetic deletion. In people who are Rh-negative, the entire Rhd gene, the specific stretch of DNA responsible for producing the Rhd protein in the first place, is simply missing from both copies of chromosome 1 that a person inherits from their parents. It isn't a different, exotic gene doing something mysterious. It's the complete absence of a gene that most other humans on Earth do have. Population geneticists understand exactly how a deletion mutation like this can become concentrated at unusually high levels within a specific population over time. particularly a population that remained relatively isolated and interconnected for many centuries, exactly the pattern seen among the Basques, whose unique, unrelated language likely functioned as a powerful cultural and reproductive barrier for thousands of years, allowing certain genetic traits, including this particular deletion, to become far more common within their community than in the wider, more genetically mixed populations surrounding them. No alien ancestry required, no hidden bloodline, just ordinary population genetics playing out over an extraordinarily long stretch of human history. But here's where things get genuinely interesting again, because even with over a century of research behind it, scientists are still actively uncovering brand new, previously invisible layers of complexity hiding inside the genes responsible for our blood types. And some of the most important breakthroughs have happened only extremely recently. In April of 2025, a team of researchers at the Blood Center of Zhejiang Province in China achieved something that had never been done before, successfully sequencing the complete ABO gene from end to end, from its very beginning, the 5' untranslated region, all the way through to its very end, the 3' untranslated region, producing a continuous 26,100 base pair sequence, the single most complete ABO gene ever recorded in the history of genetics. To understand why that achievement matters so much, you have to understand a limitation that had quietly frustrated blood geneticists for decades. Older sequencing technology, called short-read sequencing, essentially reads DNA in small, disconnected fragments, which then have to be pieced back together computationally, almost like trying to reconstruct an entire book after it's been shredded into thousands of tiny scraps of paper. This approach works reasonably well for most purposes, but it consistently struggled with certain particularly complicated stretches of the genome, including large sections of the ABO gene itself, especially a notoriously difficult region called intron 1, roughly 13,000 base pairs long, that had rarely, if ever, been successfully and completely sequenced before, despite scientists already knowing it likely contained important genetic variation. Using a newer approach called long-read sequencing, essentially reading enormous, continuous stretches of DNA all at once instead of piecing together thousands of tiny broken fragments, the Zhejiang research team finally cracked open that stubborn, previously unreadable region, and what they found inside confirmed something researchers had long suspected but had never been able to fully prove: an entire hidden layer of genetic complexity sitting inside the ABO gene that conventional sequencing methods had simply never been able to properly capture. Think about what that actually implies. The ABO blood typing system is not some obscure, understudied corner of human genetics. It is one of the single most heavily researched, most routinely tested genetic systems in all of medicine, checked on virtually every patient who ever needs surgery, every pregnant woman, every blood donor walking into a donation center. And yet, well over 100 years after Carl Landsteiner first identified it, scientists were still discovering entirely new structural patterns hiding inside the gene responsible for it, simply because the technology needed to actually read that specific stretch of DNA properly did not exist until extremely recently. It's a genuinely humbling reminder of how much of the human genome, even in areas doctors interact with constantly, remained functionally invisible until just the last few years. And it raises a genuinely fascinating question about how many other seemingly well-understood genes might still be hiding similar secrets, simply waiting for the right sequencing technology to finally catch up. And the ABO gene is far from the only blood-related genetic mystery that has only recently been solved. In 2024, a completely different research team made international headlines by finally solving a genuine 50-year-old scientific mystery involving an incredibly rare blood type known as An-Bujay. First identified in 1972, doctors had never been able to figure out exactly why some people lacked this particular blood marker entirely, a genuine medical mystery that persisted for 5 full decades. Researchers finally traced the answer down to a specific deletion inside a gene called MEL, which normally produces a protein needed for red blood cells to properly display the An-WJ marker on their surface. Without that gene functioning correctly, people simply don't produce the marker at all, becoming what's known as An-WJ negative. It's a genuinely striking reminder that even now, in an era when scientists can sequence entire human genomes in a matter of hours, there are still real unanswered mysteries hiding inside something as seemingly basic and thoroughly studied as human blood, mysteries that keep quietly getting solved, one careful genetic study at a time. So let's zoom back out now and look at the full global picture. Because blood type distribution around the world tells an absolutely fascinating story about human migration, isolation, and history, one written directly into the genetics of different populations scattered across every continent on Earth. In certain isolated indigenous communities across the Americas, type O blood reaches an almost unbelievable 90 to 100% of the entire local population. And critically, Rh negative blood is exceptionally rare or almost entirely absent among these same groups, a pattern strongly connected to founder effects, meaning a relatively small founding population crossing into an entirely new continent thousands of years ago, followed by many thousands of years with limited genetic mixing from outside populations. Compare that to parts of Europe, where Rh negative frequency climbs dramatically higher, particularly among the Basque population we already talked about, and you start to see just how dramatically blood type frequency can shift from one region of the world to another, shaped by thousands of years of migration, isolation, and reproduction patterns that most people walking around today have absolutely no conscious awareness of. Across large parts of East Asia, meanwhile, Rh negative blood becomes almost startlingly rare, in some populations dropping to less than half of 1%, a figure so low that hospitals and blood banks in these regions have to plan around it as a genuinely serious ongoing logistical challenge, since even a single patient requiring an Rh negative transfusion can strain local supply in a way that would barely register in a country where the trait is far more common. This regional imbalance becomes especially significant for international travelers and expatriates, since someone who is Rh negative and requires an emergency transfusion while traveling through a region where that blood type is exceptionally rare can face a genuinely dangerous supply gap, one that has occasionally made international headlines when blood donation networks have had to coordinate emergency shipments across borders to help a single critically injured patient. This geographic variation has enormous, very real, practical consequences for modern medicine, particularly during large-scale humanitarian crises and international disaster relief efforts. A blood bank, serving a population with historically low Rh negative frequency, faces an entirely different set of supply challenges than one serving a population where Rh negative blood is comparatively common. This is exactly why international blood donation campaigns often specifically target certain communities during a crisis, and why global disaster relief coordination for something like a major earthquake or a large-scale conflict requires careful advanced planning around exactly which blood types are likely to be in short supply in that specific region of the world. And this brings us to one of the single most dramatic real-world applications of O negative blood, a concept with genuine documented military history behind it, known as the walking blood bank. During major combat operations, when trauma casualties need immediate blood transfusions and there simply isn't time to properly test and match a patient's exact blood type under fire, military medical units have historically relied on pre-screened O negative donors kept physically present and ready near the front lines, quite literally functioning as a living, breathing blood bank capable of providing an emergency transfusion within minutes rather than hours. This practice dates back to major 20th century conflicts and continues in modified, modernized forms within military medicine today, precisely because O negative blood remains the single safest option when doctors are forced to transfuse a patient without the luxury of confirming their exact blood type first. Every single soldier serving in a unit like this, screened and ready to donate their own O negative blood at a moment's notice for a wounded comrade, represents a genuinely striking real-world example of exactly why this specific, relatively rare genetic trait has taken on such outsized importance in modern emergency medicine. Modern battlefield medicine has actually refined this concept even further in recent years, developing what's sometimes called low-titer O, negative whole blood protocols, where pre-screened donors with especially low levels of certain antibodies are specifically identified in advance and kept ready to provide whole blood transfusions that are safer and more effective than standard blood products in a genuine trauma emergency. Military medical researchers have found that whole blood transfusions, delivered as close to the point of injury as possible, dramatically improve survival rates for severely wounded soldiers compared to waiting until they can be transported to a fully equipped hospital. This entire modern approach to combat trauma care traces directly back to that same basic genetic fact we've been circling throughout this whole story, that one specific, relatively rare combination of missing surface proteins on a red blood cell can mean the difference between a soldier surviving long enough to reach proper medical care or not. Given just how critical and just how chronically scarce O negative blood consistently turns out to be, it should come as no surprise that scientists have also been working hard on an entirely different kind of solution, manufacturing it directly in a laboratory. In 2023, a team of researchers in China published a study describing exactly how they successfully used CRISPR gene editing technology to engineer human stem cells capable of producing O negative red blood cells from scratch. Starting with human-induced pluripotent stem cells, cells that can be coaxed into becoming almost any cell type in the human body, the research team used a precise CRISPR-based technique to deliberately introduce a premature stop signal directly into the RHD gene, essentially recreating, in a controlled laboratory setting, exactly the same kind of genetic deletion that naturally occurs in people who are Rh negative. The resulting engineered cells were then carefully guided through a multi-stage differentiation process until they developed into fully mature red blood cells, ones that tested negative for the RHD antigen, and showed no reaction whatsoever when exposed to anti-RHD testing reagents, essentially artificial, laboratory-grown O-negative blood cells. This kind of research sits at the very edge of what's sometimes called regenerative transfusion medicine, an entire emerging field built around the ambitious goal of one day producing safe, reliable, laboratory-grown blood products at a scale large enough to actually supplement, or even someday reduce, dependence on traditional human blood donation. The appeal is obvious. A laboratory producing engineered blood cells doesn't get tired, doesn't skip a donation appointment because of a busy week, and doesn't experience the kind of unpredictable seasonal dips in donor turnout that have directly contributed to real-world shortages like the ones the American Red Cross has now had to publicly declare not once, but twice. While this technology remains firmly in the research and development stage rather than something available in hospitals today, it represents a genuinely fascinating potential solution to a very real chronic global shortage. The possibility of one day manufacturing a reliable, laboratory-grown supply of the exact blood type doctors reach for first in the most desperate emergencies, without needing to rely entirely on the unpredictable generosity of human donors walking through the door on any given day. Step back now and look at the complete picture this entire story paints. Type O, negative blood, is not some mystical ancient bloodline connected to a hidden branch of humanity, and it's not evidence of anything remotely extraterrestrial, no matter how persistently those particular theories continue circulating across the internet. It is the direct, well-documented result of a specific genetic deletion, the complete absence of one particular gene, a trait that happened to become concentrated at unusually high frequency within certain historically isolated human populations, particularly the Basques, purely through ordinary population genetics, playing out over thousands of years. But the actual unglamorous scientific explanation behind O-negative blood doesn't make the story any less remarkable. If anything, it makes it more remarkable, because this one specific genetic quirk, essentially a small ancient accident buried inside human DNA, has ended up sitting at the very center of modern emergency medicine, quietly saving countless lives in operating rooms, ambulances, and battlefield medical tents around the entire world every single day. And that brings us right back to where we started, that unprecedented national blood crisis, declared not once but twice now by the American Red Cross, both times driven specifically by a dangerous shortage of type O blood. Somewhere out there today, a hospital is running low. Somewhere out there today, a doctor is quietly hoping the next patient wheeled through the emergency room doors happens to match whatever blood is currently sitting in the refrigerator down the hall. And if that patient's own blood type is unknown, or if there simply isn't time to check, it's O-negative that gets reached for first. That one specific genetic combination, the absence of two separate proteins on the surface of a person's red blood cells, has quietly become one of the most important, most consistently needed resources in the entire modern medical system. Precious specifically because it is rare and irreplaceable. Specifically because, even after more than a century of genetics research, scientists still cannot manufacture it fast enough or in large enough quantities to keep up with how desperately it is needed. There's something genuinely worth sitting with in that contradiction. We live in an era where scientists can sequence an entire human genome in a single afternoon, edit specific genes with surgical precision using CRISPR technology, and grow functioning human blood cells inside a laboratory dish. And yet, when a hospital runs critically low on O-negative blood, none of that extraordinary technology can instantly solve the problem. The solution still comes down to something almost old-fashioned by comparison, an ordinary person choosing to walk into a donation center and roll up their sleeve. Every single unit of O-negative blood currently saving a life somewhere in the world right now came from another human being who simply decided to show up and give it. So if you happen to be one of the small percentage of people walking around today carrying O-negative blood inside your own body, you are quite literally carrying one of the rarest, most consistently sought-after genetic combinations found anywhere on the planet, not because of some hidden ancient bloodline and not because of anything remotely mysterious or otherworldly, but because of a very real, very well-documented genetic deletion that happened to be passed down to you through your own family line, generation after generation, stretching back through your own personal, entirely human history. And the next time, scientists managed to crack open yet another hidden layer of complexity buried inside the genes responsible for your blood type, exactly as researchers did in 2025 when they finally sequenced the complete ABO gene for the very first time, or exactly as they did in 2024 when they finally solved a genuine 50-year-old blood type mystery. Remember that even something as seemingly simple, as thoroughly studied, and as ordinary as your own blood type still has entirely new chapters left to reveal. The truth was never hiding in ancient aliens or secret bloodlines. It was hiding in plain sight, quietly encoded inside your own DNA the entire time, waiting for the right scientists with the right technology to finally read it properly.